we now have an offline talk, and the speaker is where? Just run for it. If you are the speaker, run. Run. Sorry, it's Alina Mikhailova from the Baltic Federal University in Kaliningrad. Yes, sorry.
Thank you. And thank you to Eugene Koonin and Tver Kondrashov for great science. Can I remove the mask, maybe? Okay. And now I would like to talk about mitochondrial evolution and in particular talk about how mitochondrial mutagenesis can shape amino acid substitutions. And it has been shown that during long-term evolutionary processes, some amino acids can disappear, and we call them losers. And some of them can increase their frequencies, and we call them gainers. And in this research, authors described the global trend of this amino acid shift, but they didn't give any explanations what is the reason of this trend. Is it mutagenesis or is it selection? And this is why we decided to prove this hypothesis on mitochondrial DNA, because we know rules of mutagenesis of mitochondrial DNA, and so we can determine role of mutagenesis in this trend, and we also did it. So let's look at mitochondrial molecule, mitochondrial DNA molecule. This is a circular molecule with asynchronous type of replication. This means that one strand spent more time being single-stranded, and this condition is quite mutagenic for DNA. So in case of mitochondria, this strand is called heavy chain, and we expected that on this chain can occur more substitutions, and we have found that there are some substitutions. Moreover, we have found that mitochondrial mutational spectrum is quite asymmetrical, with two most common transitions, A to G and C to T. But further, we will call them G to A and T to C in their complementary or light chain notation, because light chain notation better represents mRNA and can be translated directly into amino acids. And so let's take a look which is the effect of these transitions. At first, it has been shown that codon usage in mtDNA can be shaped by its asymmetrical mutagenesis. And for more information about this study, you can visit poster of Eldar Haybulin, and we go on. So we expected that something similar should happen with amino acid substitutions. And let's take a look on amino acid content, taking into account these transitions, which have to create some specific pattern. And according to our hypothesis, we expect that amino acids should mutate from the external columns of this mitochondrial genetic code table towards middle, following the trend of transitions. So from G to A and from T to C. And those amino acids which are located in the corners of this table we consider as losers. For example, they are presented with valine, glycine, cysteine, etc. And those amino acids which are located in the middle of this table we consider as gainer, and they are expected to increase their frequencies. And so we tested this hypothesis. For example, we expected that substitutions from valine to alanine have to be more frequent than reverse substitutions from alanine to valine, the opposite types of substitutions. And we took several data sets. And the first one was the data set of human cancer mutations. And what we did exactly. For example, take a look on this row with substitutions from valine to alanine. We observed 78 substitutions from valine to alanine, and then we normalized these substitutions considering the number of valine. And here it's 136. So we divided 78 by 136 and obtained number 0.5. And this number is the rate of expected substitutions. And then we did the same with the opposite substitutions from alanine to valine. And we obtained the rate of unexpected substitutions. And here it's 0.1. And already we can see that the trend, the frequencies, the rate of expected substitutions is 5-fold higher than rate of unexpected substitutions. And then we did this analysis on other substitutions, and additionally added several data sets to cancer data set. It was human pathogenic mutations from MitaMap database and human polymorphisms. And what we can see, we get the odds ratio for these substitutions, and we do see the shift of the rates of expected substitutions. So that means that the rate of expected substitutions is more prominent than the rate of predominant than the rate of unexpected substitutions. And additionally, it's interesting that it also corresponds, the shift in cancer data set is the highest, and it corresponds to its relaxed level of selection. So the level of selection in cancer is more relaxed, and the shift of the rate of expected substitutions is also high. And for more details on this study, please visit the poster of Alina Mikhailova. So we can see that there is a universal trend in human population, and we decided to test this trend in other taxa, and we counted number of gainers and losers in different taxa, becoming from fungi, plants, and ending with mammalia and aves. And what we expected was that the highest number of gainers should be in species with lowest effective population size, because in low population size mutations can accumulate, then can fixate there. And then we additionally we added to the analysis genomes of alpha proteobacteria, which is considered as closest relative to protomitochondria. And we observed there that the fraction of gainers is there low, but we do know that population size of alpha proteobacteria is quite high. And this is in line with our hypothesis that the excess of gainers and the deficit of losers are driven by mutational processes, and these substitutions are fixated in species with low population size. And for more details on this study, I recommend you to visit poster of Valeria Lobanova. And to conclude, I can say that it's interesting topic to research, and the understanding of amino acid substitutions can help to understand, for example, pathogenic mutations in human mitochondrial DNA. And that's it. And thank you for your attention. Thank you, the organizing committee. And I'm ready for your question.
Okay, we have time for one question probably.
Can you hear me? Thank you for the talk. Prolines and glycines are usually considered as the most conserved amino acid because they are important for structural features. So for me, it seems to be quite unexpected why these residues are in top 3 of losers.
So, as I said, of course, all these substitutions, they are non-synonymous, and they can be slightly deleterious. And it may be explained by the fact that they are fixated in populations with low effective population size, which help to fixate the deleterious mutations. And this can be explanation why this disappearing of these substitutions emerge. And briefly, we are degenerating because we have lowest population size compared to, for example, bacteria.
Okay, thank you. We definitely cannot do better than this last statement. I wholeheartedly agree. Thank you.
In terms of genetics, we are...
In all terms. Okay, thank you.